So this is a lecture on diversity of both viruses and bacteria for Bio 101 for August 26, 2020. And so for this portion of the lecture, we're basically going to focus on the viruses, and then there'll be a part 2 that focuses on bacteria. So the specific objectives that are covered in this lecture are here. We're going to define some terms, and then we're also going to talk about if viruses are living and non-living, as well as what a viral spike protein is and what it does. And so some virus basics. Viruses are acellular or not alive, right? So in order to be alive or considered living, an organism needs to be made up of at least one cell. And technically, since viruses are acellular or without a cell, they are not alive. And that's because of some of the properties we talked about in a previous lecture, right? They are unable to replicate themselves. They are unable to grow. And so they don't have all of these kind of like properties of a living thing. They also do not have any cells. However, viruses can exist in two different states. They can exist outside of a cell. When they're in that state, they're technically inactive. Once they have bound to and inserted themselves inside of a cell, they can also exist that way. And they, once inside of another cell, will multiply. And they can only do that when they use the resources of an additional cell. As viruses lack their own metabolism and their own ability to reproduce and grow, as I previously said. So viruses can exist outside of the cell. When they are outside, they're inactive. Once they have infected or gone inside of a cell, they can hijack that cell's machinery to multiply and make more viruses. And viruses are infectious agents that cause major diseases, as we all know right now, living through this COVID-19 pandemic. And viruses are not only able to infect humans, but other animals and plants as well. And viruses can actually also infect bacteria. So depending on which type of virus, it can infect different cell types. Some viruses can infect every type of cell. Some viruses can only affect bacteria or only plants or only humans. And we'll talk a little bit about how that works when we get to the viral spike proteins. And so viruses, as we said, are infectious agents, and they can cause many different viral diseases. And I'm just going to discuss some diseases of different sort of transmission routes or different routes that they can use to infect human patients, right? And so the first group I'm going to talk about are airborne viral diseases, where the viruses are transmitted through the air in respiratory droplets. Some examples of these diseases can be seen here, listed on the left, and then the viruses that cause each of these diseases is listed in the right. And some of the common features associated with an airborne viral disease are that it causes what's known as an acute infection or a short-term, very harsh infection. Most airborne diseases are also highly contagious and infective, and you can imagine that it's because of our high dependence on oxygen and needing to breathe the air. An airborne viral disease can travel pretty far, and it can also travel easily between two people. And these are just some examples of two patients who have contracted airborne diseases. And so this person here has chickenpox, this person here has smallpox. Both of these diseases are sort of characterized by these rashes or these blisters and pustules that will form on the surface of the skin. You may have never seen a patient with smallpox before. It's been effectively eradicated from the United States. But those blisters are actually highly contagious as well. And one thing to note about particularly COVID-19 in particular is that when these viruses travel using respiratory droplets, the respiratory droplets can only get so far. And in the case of the SARS-CoV-2 virus, it travels in droplets that can only make it about 2 meters or 6 feet from an infected patient out into the air. And so that is where that sort of 6 feet physical distancing guideline comes from. the fact that SARS-CoV-2, once it reaches past 6 to 7 feet, it's not going to be able to be transmitted as effectively through the air. And so if you give yourself a 6 feet barrier, it makes it much harder for SARS-CoV-2 to infect you than if you don't. Some diseases or viral diseases are caused by direct contact, which is sort of like person to person or person to animal contact. And some examples of those can be seen here. So AIDS is one that you may have already heard of. It's caused by the HIV virus. Herpes and mononucleosis are two others that are caused by direct contact, both caused by these viruses listed here. And what's interesting about the direct contact diseases is that rather than a short-term strong infection, direct contact viral diseases like AIDS, like herpes, can lead to persistent infections over the course of a patient's lifetime. And so these viruses can live within the cells and sort of stay dormant, having a very slow progression of disease. And a patient may be infected with these viruses and be asymptomatic or not know they have them for a very long time. And then there are certain things that might sort of turn that virus on and activate it and cause it to lead to actual symptoms of infection. And that can happen consistently over the period of a person's life. And so one thing that you may have heard of, and at least in terms of maybe herpes or the genital warts caused by human papillomavirus or HPV, is that there's these flare-ups. And so the virus is already inside of you, possibly latent, living in your cells, kind of dormant, and then something activates it to replicate and cause an infection that you can see. And that would be like the flare-ups that people talk about. And then some viral diseases are transmitted from animals to humans, specifically through contact with a particularly infected animal. And two examples of a zoonotic or an animal-borne disease are Ebola, caused by the Ebola virus, and rabies, which is caused by rabies virus. And some common features of the zoonotic or animal-borne diseases are that the patient needs to come into contact with an animal for that disease to originally begin. And animals can serve as what's known as a reservoir for these viruses. And so the animals can have the virus living inside them, but it won't affect or kill them. And so they're effectively just a reservoir or like a holding tank for potentially infective viruses. These viruses and these diseases have what's known as a long incubation period, or a long time between when you're first exposed to the virus and when you get symptoms. But once you get those symptoms, there's a very quick disease progression. And so there's a long kind of incubation period when you've been infected with the virus and you don't see any symptoms. And then once you do see symptoms, the disease progresses to death very, very quickly. In the case of this patient over here, or these patients, they all have Ebola virus, which is a hemorrhagic fever. And hemorrhagic means bleeding, right? And so you can see in all of these different images, there is bleeding from different orifices, your mouth and your ears, as well as your eyes. There are bloody pustules associated with Ebola virus, and Ebola patients are highly infective. So this blood can cause transmission of Ebola virus from patient to patient, and it can also Ebola can also be transmitted very easily through digestive matter, so like fecal matter or through vomit. And so Ebola has a high transmission rate between patients and their caregivers because it is so infective. But as I said before, Ebola virus can live very well inside an animal reservoir, and you'll never know that it has any of these symptoms because that virus can multiply there, but it doesn't actually cause symptoms until it reaches a human patient. And so there are a lot of viruses that can infect humans and cause disease, and you see some listed here. And some of these diseases like smallpox, cholera, and polio are no longer commonplace, at least in economically developed countries, including the United States. And And the reason for that is vaccination. But it is important to remember that it is not just the United States in this world, and there are some places where some of the more intense viral diseases still exist. Polio, for example, still existed in Africa up until about several days ago, and there was a massive vaccination drive a few years ago that has now rid Africa completely of wild polio virus, which is actually some awesome news in the midst of 2020. And so vaccination is as close to a medical miracle as we have ever come. And the cases of a disease before and after vaccination dramatically decrease, almost to 100% in the case of certain diseases. And so we'll talk more about vaccines and how they work later in the class, but it's important to realize that the viral diseases can be treated effectively with vaccination and almost be kind of removed from the world. And so virus structure can vary between different viruses. So you can see here sort of a to scale drawing of a bunch of different viruses, and you can see there's a lot of different shapes and sizes there. And a complete virus particle is what's known as a virion. Those virions vary in size, but they all contain the same two things. They all contain a nucleic acid to encode the genetic information. It might be DNA, it might be RNA, but there's always a nucleic acid in a virion. And the other thing all virions have is a capsid, or coat made of protein that basically wraps around the nucleic acid and keeps it safe and protected. And that's it. That's all viruses have. They are basically protein sacs filled with DNA. Some of them occasionally will have an additional layer on the outside known as an envelope, and that envelope is made of lipids or fats, and it wraps around the capsid on the outside. So you can see a virus with no envelope here on the left. It's just nucleic acid and that protein coat, whereas this envelope virus on the right has nucleic acid, capsid, protein coat, and then this fatty envelope layer on the outside. And what you see here are these little green spikes sticking out of the envelope, which are actually really essential to the virus being able to replicate and it also infect other cells. And so we're going to talk a little bit more about that on this slide. And so a viral envelope you can see here in this dark maroon, or actually around this virus there. As I said, it's a membrane structure. It's made of lipids or fats, and it's got some sugars embedded into it as well as some different proteins. And the most important protein that's embedded or stuck into that viral envelope is what's known as a spike protein. And so the spike proteins can be seen here in the rabies virus as these little sort of club-shaped red structures. They can be seen here on the SARS-CoV-2 virus on the outside, and then another coronavirus down here. You can see them on the outside of those viruses. And the spike proteins are particular to each virus, so each different virus has different spike proteins, and what they do is they help that virus or that virion attach to a host cell and ultimately infect it. And you can use the spikes to identify viruses because they vary in shape, they vary in the proteins that make them up, and so by looking at the spikes of a specific virus, you can identify which one you're looking at. And in addition to being able to use them to identify viruses, you can also, spikes also determine what types of cells a virus can infect. Some spike proteins are able to bind to a bacteria and infect that cell. Some are able to bind to a lung cell specifically. Some are able to bind to a brain cell, and those infect those. And that's where that specificity of who and what cells get infected comes from. It comes from the spike proteins. And a lot of research right now into SARS-CoV-2 and how to fight it is looking at spike proteins and how to prevent them from binding to human cells and allowing the SARS-CoV-2 virus to infect us.